Welcome to Online Lecture 4, which covers Chapter 6. This lecture is recorded in 3 parts, so please make sure to watch and take notes on all 3 of them. The topics covered in this chapter are the pathways that harvest and store chemical energy in living organisms. We already started to look at metabolic processes in Chapter 2 and in the lab. In the lab, we're studying the fermentation reaction, which is depicted here in the right-hand corner of your screen. These are fermentation reactors that are involved in industrial processes. In this case, the purpose is to generate ethanol for human consumption. In cells, metabolic pathways are expressly for the purpose of harvesting and storing chemical energy. As we saw in Chapter 2, organisms cannot create or destroy energy. Yet they have a constant need for energy to fuel cellular processes. So the focus of Chapter 6 is how organisms obtain the energy they need and change it into a form that's useful to the cell. Try to keep this overall picture in your mind as you go through this chapter. The chapter has a lot of detail that I don't expect you to know. So as I go through the chapter and in your reading, please try not to get too hung up on the details, but to instead keep focusing on the big picture. And the big picture is listed in the learning outcomes on page 3 of your syllabus, learning outcome 3, which says energy transformation pathways and metabolism, with a focus on glycolysis, fermentation, cellular respiration, and photosynthesis. We're not going to be memorizing any of those in tremendous detail, but instead, we're going to be looking at this from the standpoint of how it is that organisms obtain the energy that they need and transform it into a useful form. Hopefully, the work we did in Chapter 2 has given you a pretty good idea of how these energy needs might be met. From that chapter, you should recall that chemical reactions are coupled. This coupling occurs in the way that is shown in this diagram, in that the energy that is released from exergonic reactions is then used to drive an endergonic reaction. For example, the synthesis of ATP. That ATP contains a lot of energy, and when ATP is hydrolyzed to ADP, it can be coupled to an endergonic reaction, which requires the input of energy. And as we go through this chapter and discuss various kinds of coupled reactions, it's very important for you to keep in mind that each of these reactions is catalyzed by a different enzyme and actually occurs separately, even though they're coupled. They do not occur simultaneously in the active site of a single enzyme. Remember, each enzyme has a specific substrate. So coupled reactions would occur in close physical proximity to one another, and there would be an energy exchange among them. But there are separate enzymes catalyzing each of those reactions. Now this figure shows one of the most biologically important coupled reactions, those involving ATP. We've mentioned ATP several times today already, but also in Chapter 2, and we're going to take a closer look at this molecule today. The term ATP stands for adenosine triphosphate, and we focus a lot on this ATP because it's a kind of energy currency in cells. It can harvest energy that's released from exergonic reactions or store energy in its bonds, and then be transported to another place where it can release that energy to drive a cellular reaction. So just how does ATP store energy? In its chemical bonds. This slide is showing the structure of ATP. You'll see that there is a sugar here, a pentose sugar, ribose, which is attached to 3 phosphate groups, and also to a nitrogen-rich compound called adenine. Look at this very carefully. Does this look to you like anything that you've seen before in this class? If you've said a nucleotide, you're spot on. What? You might be thinking, Does this mean that the ATP that drives cellular reactions is the same A or adenine that's in our DNA? Yes, I mean exactly that. And get this, it's the hydrolysis of this ATP that actually drives DNA synthesis. We'll learn more about this process of DNA replication in a later chapter, but for now, just sit and think for a minute about how amazingly efficient cells are to build DNA out of the same molecule it uses as an energy currency. Not only does ATP release energy when it's hydrolyzed, but it readily exists in the cell in three different forms. ATP refers to the triphosphate form. When one phosphate group is lost, the molecule is referred to as ADP, that stands for adenosine diphosphate. And when the second phosphate group is lost and there's only one remaining, the molecule is referred to as AMP. AMP is actually an important messenger in cells. If you take a cell biology class, you'll learn about that. It's really beyond the scope of this class. But suffice it to say that cells are extremely efficient and do use molecules for very many purposes. So let's focus now on the role of ATP as an energy currency molecule. So why exactly is it involved in so many coupled reactions? Recall from Chapter 2 that the change in free energy associated with ATP hydrolysis is negative 7.3 kilocalories per mole, which of course means that in this case the free energy of the reactants is greater than that of the products. So let's take a look at the chemical structures of the products and the reactants to see why this is. This slide is showing ATP at the top. Here's the nitrogenous base and the sugar, and then the three phosphate groups. Everywhere you see these squiggly lines indicates what we call a high energy bond. During ATP hydrolysis, one of these high energy bonds is broken and two new bonds are formed to form this hydroxyl group here and here on the two phosphate groups. So the free energy of the reactants is greater than the free energy of the products because the energy contained in this phosphate bond is higher than the energy in those hydroxyl bonds that form after hydrolysis. Therefore, when ATP is hydrolyzed, free energy is released, which of course is a great means for providing the fuel to drive cellular reactions. The opposite of this reaction, ATP synthesis, is a very effective way to store energy. And this is why ATP is involved in so many coupled reactions. However, ATP is not the only molecule that can transfer energy in coupled reactions. There are actually many. Another very important example of reactions that involve energy transfer are oxidation reduction or redox reactions. In redox reactions, energy can be transferred by the transfer of electrons. A reduction reaction involves the gain of one or more electrons and is an endergonic reaction, whereas an oxidation reaction involves the loss of one or more electrons and is an exergonic reaction. Redox reactions always occur together. One compound is always oxidized while the other is reduced. So in this example, compound A, which is losing electrons, is being oxidized through an exergonic process, whereas compound B, which is gaining electrons through an endergonic process, is being reduced. Here's a mnemonic to help you remember what happens in a reduction and what happens in an oxidation. And the mnemonic is GER-LEO, where GER, or G-E-R, stands for gain equals reduction. So this is the gain of electrons equals a reduction. And LEO stands for loss equals oxidation, where the loss of electrons equals oxidation. So redox reactions then are always coupled. Some redox reactions involve only the transfer of electrons, while others involve the transfer of a hydrogen atom and an electron. Because of this, atoms with fewer hydrogens are more oxidized, whereas atoms with more hydrogens have more bonds, are more reduced, and store more chemical energy. Let's look at an example here of 5 carbon compounds, each of which only contain a single carbon atom. These 5 compounds are in various states of oxidation. On the left is methane, the most reduced 1-carbon compound with the most chemical energy stored in its bond. You'll notice it has 4 hydrogens bound to that central carbon atom. Compare that to carbon dioxide, shown on the right-hand side of the screen, which is the most oxidized form. It has no hydrogens, and interestingly enough, carbon dioxide is often the final waste product of metabolic processes, because this carbon dioxide represents a fully oxidized compound, and there's not enough energy left to harvest from it to bother with metabolizing it any further. Thus, it's usually released as waste. Therefore, another way of storing energy then is to reduce compounds, and a way of releasing it to drive cellular reactions, for example, is to oxidize compounds. The next energy carrier we're going to consider is involved in just this kind of reaction. The compound is called nicotinamide adenine dinucleotide, or NAD for short, in its oxidized form, or nicotinamide adenine dinucleotide hydride, or NADH, in its reduced form. This coenzyme, as it's called, is a very important electron carrier in redox reactions. So let's take a look at its chemical structure. Here we have the oxidized form of the compound, NAD, and when this compound is reduced and gains a hydrogen atom and 2 electrons, it forms NADH, the reduced form. And these differ in the presence of this hydrogen atom here and the loss of the charge on the nitrogen atom. That, of course, is an endergonic reaction because it's a reduction reaction. NADH then can readily be oxidized to NAD in an exergonic reaction in which energy is released. If this looks familiar to you, it should. This is very similar to what we discussed with ATP hydrolysis and ATP synthesis, how you have a reversible endergonic and exergonic reaction that can be used to either harvest energy in a reduction reaction and carry it to somewhere else in the cell, and then release that energy in an oxidation reaction. So ATP and NADH both have something in common as energy carriers. And I want to call your attention now to the structure of NAD. If you look closely at NAD, you'll see there is our pentose sugar, ribose here, which is attached to an adenine, and then to another group called a nicotinamide. This structure should look familiar to you, and hopefully you can see that ATP forms a backbone which can readily be modified to make this NAD. So we just have another example here of cells using compounds without reinventing the wheel for a lot of different purposes. So the few purposes we've seen for ATP already are involved as an energy carrier. It's a structural part of DNA, and we see from this example that it can be modified to yield other useful compounds. And I also just want to mention on the right-hand side of your screen here where these dashes are, that's meant to indicate that the rest of the molecule on the right-hand side here is just the same as what's on the left. They just haven't drawn it for the sake of simplicity. This slide is showing the chemical equation for the reduction of NAD to NADH, and at the bottom, the oxidation of NADH to NAD. These reactions involve much more energy transfer than ATP, where the change in free energy was negative 7.3 kilocalories per mole for ATP hydrolysis. In comparison, the change in free energy for the oxidation of NADH releases about 50 kilocalories per mole. So this is a much higher energy compound than ATP. And what we're going to see is that in cells, the NADH-NAD system transfers more energy than ATP, but they actually work together. The NADH is used to trap energy in large amounts, which is then transferred to ATP. And then the energy to drive the anabolic processes needed by the cell is actually fueled in little packets by ATP. Of course, the energy to form this ATP is often derived from NADH. However, unlike what we've seen before, these processes are not directly coupled. Just think about this and it'll make sense to you. There's much more energy released when NADH is oxidized, so 50 kilocalories per mole for this reaction that's shown here at the top of your screen, whereas only 7.3 kilocalories per mole is necessary for the reaction at the bottom of the screen to take place. So if these were directly coupled, there'd be a tremendous waste of energy. Instead, a system has evolved that couples the energy release of NADH oxidation to ATP synthesis, and it does so in a very efficient manner. This manner is called chemiosmosis. During chemiosmosis, protons are pumped in between two cell membranes, and they get stored up in there, kind of like water behind a dam. Then, the energy that these stored up protons release when they flow across a membrane is used to create ATP. So what's happening during chemiosmosis is that potential energy in the form of what we call a proton gradient ends up stored as chemical energy in ATP. So think of it this way, as water turning the turbines of a dam. So the proteins are flowing through the membrane, through a special protein system, which we'll talk about later, and they're turning the turbines of an enzyme that makes ATP. This slide shows a diagram of this process. What you're looking at here is a lipid bilayer. This is a membrane. In the eukaryotic examples that we're going to consider during this lecture, this membrane would actually be part of the mitochondrion. And what happens during chemiosmosis is that protons get pumped across this membrane, and they eventually accumulate on this side. So you can see lots of protons over here. This is our water that's being stored up behind the dam. And then eventually, when enough protons are stored up here, they actually flow back in the opposite direction through a special protein called ATP synthase. So this ATP synthase then catalyzes the synthesis of ATP from ADP and inorganic phosphorus. This slide shows a close-up of ATP synthase, the enzyme responsible for ATP synthesis during chemiosmosis. How is this process linked to NADH? Well, it turns out that it is NADH that provides much of the energy to generate this proton gradient across the membrane in the first place. We're going to look at this process of synthesizing ATP through chemiosmosis in some detail. But before we do that, we really need to consider where this NADH is coming from. It's energy currency, but it's not a major food. Instead, we consume things like fats and proteins and carbohydrates, and these must be converted to NADH and then ultimately to ATP through chemiosmosis. I want to stress here that the metabolic processes of cells are extremely diverse. In the next section, we're going to look at a universal example that begins with glucose. But please remember that there are many other potential energy sources. Just think about all the different ones we're going to use in the fermentation lab. This should also help you focus on the overall picture rather than the details of the processes that are covered in the metabolic pathways we'll discuss in the next two online lectures. In response to requests from some members of the class, I'm going to pause here and tell you what I think are the major concepts from this first lecture that I've given for Chapter 6 here. So the major concepts of this section include the energy coupling process and how it is that inorganic and exergonic reactions are coupled and used to drive cellular reactions. You should be familiar with the structure of ATP in all its various forms, meaning ADP or AMP as well, and the energy changes that are associated with those different forms of ATP. You should be familiar with redox reactions and the role that they can play in harvesting and transforming energy in cells. You should be familiar with NADH and NAD and that redox reaction. Finally, you should know that NAD is used to drive much of the ATP synthesis in the cells. I recommend, if you're unclear about any of these processes at this point, that you stop and review this lecture, possibly your textbook, and your notes before you go on to Part 2 of Online Lecture 4.